Okay, so good morning, everyone. Today's speaker is Matan Mussel from the University of Haifa. Matan is a new faculty member there, the Department of Physics. This is also a new department for Haifa. I know Matan a long time ago when he was a postdoc at the NIH. We were neighbors back then. So I'm happy to see you here. And your talk is about steep transitions in soft materials and corresponding biological aspects. So the stage is all yours, and thank you.
Thanks a lot for the invitation. I was really looking forward to come and visit you in person and hear a bit about your own work. Unfortunately, yeah. I will go and visit Udi and hopefully some of you at some other time. Right, so I'll share my screen and okay, so you should now see it, I hope. Right, so let me start with mentioning that indeed the University of Haifa officially approved a new department of physics just 3 months ago, more or less. And I will just start with 2 minutes of advertisement. Starting next October, we will offer a new bachelor's program in biophysics. So if you have kids or friends or anyone you think who enjoys math and physics but is interested in problems that are related to biology or medicine, we built a program which is aimed to couple a broad education in biology, physics, and mathematics. There is no similar program currently at Israeli universities. There was at the Bar-Ilan University something similar, but they closed it down last year. So right, so that's about that, and now we can go to science. I'm doing partially theory, partially relatively simple experiments. My interest is living cells. What drives my philosophical approach is how can we quantify certain functions in living cells into a mathematical formulation based on physical and chemical principles. Specifically, how can we write down models that have a predictive power? And in biology, as probably all of you are well aware, it is rather challenging because every molecule counts. The number of components and all of the nonlinear couplings is huge, makes the problem very incomprehensible. And just to give one example among many, so here I just plot a cartoon of a certain process. This is a process of vesicle fusion into the cell membrane. So all of these yellow spheres that you see here, they represent lipid vesicles that exist inside living cells. Their diameter is about 50 nanometers. And under certain conditions, they can move into the cell surface and fuse into the membrane, which kind of envelopes the cell body, and thus spilling their content into the extracellular solution. So every word that you see here in English, it represents a certain molecule, a certain protein, which have an important role in part of this process. And you can imagine that writing down equations that are based on physical principles that account for each and every molecule and how it interacts with one another, it's rather complex and challenging. And to clarify the problem, we can look at different computational approaches and their predictive abilities. So suppose that we really want to account for each quantum mechanical interaction between different particles, or even coarse grain a bit and use some kind of effective classical principles like Newton's second law, electromagnetism, certain phenomenological chemical interactions like van der Waals. The best that we can do with today's abilities is to write down simulations that can model maybe 10 or 100 nanometer cubed and then make predictions over time scales of nanoseconds, maybe 10 or 100 nanoseconds, if we are willing to wait a week or a month for the computer to finish the computation. And if we're interested in full cells with size of a micrometer or even tissues which have sizes of tens of micrometers, millimeters, and even larger length scales. If you're interested in processes that last milliseconds or seconds, etc., then these molecular computational approaches are just insufficient today, and I'm not even getting into the number of unknown parameters that we need to feed the model. The alternative approach is to try and use highly mean field or coarse-grained models from the field of continuum dynamics. And of course, the problem with this is that we know that every molecule counts, right? So when we really coarse grain so much, we're ignoring so many molecular details, and the question arises, is this feasible or how useful is it? So the philosophy is perhaps to try and identify certain aspects in cells where it seems legitimate to describe the properties of the material, the properties of the cell using continuum principles, and then maybe as a second order, introduce how different molecules modify the parameters of the phenomenological description. And just to give you one of the most famous examples in this philosophy of biological physics, one can write down a relatively simple expression describing the energy of the cell surface. And we account here for the surface tension, that's sigma here, or the rigidity of the material, that's kappa, or the pressure difference. And for different numbers or values for these physical parameters that can independently be measured, we can calculate the minimization of the energy and thus extracting different types of shapes. And these shapes, interestingly, really correspond to experiments made on lipid vesicles, as you can see in the upper figure. And if we look from right to left, we can see that this relatively simple theory captures the essence of this idea of vesicle fusion into the cell membrane. So what we have now is a mathematical formulation, and instead of just describing this cartoon with words and kind of English description, we can now ask the question, how do different molecules not promote vesicle fusion, but rather how do they influence the macroscopic properties of the material? How do they modify the surface tension or the rigidity, thus promoting vesicle fusion? So that's kind of the general idea. Where can we identify aspects that we can describe as more coarse-grained, and then later introduce the molecular details? So I will try to describe three processes. My goal was not to go into the full details of one story, but rather to introduce the general things that I'm doing. And maybe one of you is working on something similar, or we can find some interest or mutual collaboration or something. And if not, you're just going to hear some nice stories. So I'm going to talk on three material properties that include some steep transitions. And the question is, did nature find use for these physical changes in order to produce some biological functions? So gradual or minute changes in different parameters can lead to significant changes in the material. The first one is called the Perling instability. This is a behavior which dates back to works of Plateau and Riley 150 years ago, where we can look at soft cylinders. In some cases, they are rigid enough to maintain a stable cylindrical shape, but in other cases, the surface tension is high enough to promote a shape transformation of these sinusoidal dilatations and constrictions, where the surface area, the surface tension tries to minimize the surface area. This is what you actually get in water every day when you take a shower, and the cylindrical column of water forms these kind of different separate droplets. And the second phenomenon I'm going to discuss is that of sound waves, namely compression and rarefaction changes in density that propagate within a medium. In my case, I'm going to talk about sound waves that propagate within a lipid membrane, and I will ask the question, what happens if these kind of local compressions drive a local phase transition in the membrane? What kind of interesting properties arise for these pulses? And then if I will have a few minutes to spare, I will briefly discuss the idea of a sharp transition or a steep transition in the volume of charged polymer, cross-linked polymers or gels as a response to competitions of different ions, mono and divalent cations. In my case, and this is actually highly related to the story of superabsorbance. So yeah, all right. So I'm going to start with the phenomenon of purling. In the biological literature, you can see the words beading or varicose for different situations. And this is a rather universal phenomenon for soft materials. Here's an example found in pure liquid, where when the surface tension is sufficiently high, it drives this shape transformation with a very thin region here called the tether connecting the two droplets, or it can fully break, thus having separate droplets. We can find it in soft cylinders made of different types of gels, in cylinders made of pure lipid membranes, and also this was identified in different conditions in biological cylindrical protrusions of cells here, driven by increasing the tension in axons, which are part of brain cells of neurons. Here is another example where one axon becomes a kind of pearl when changing the osmolarity of the extracellular solution. And in this interesting case, the cell was able, after a few minutes, to recover back to the original stable cylinder. So there were some internal changes, or by different chemical disruptions that interfere with the internal rigidity of the cell protrusion, in this case, disrupting the actin, which is part of the polymer skeleton that increases the rigidity of the cell solution. Okay, so this is, as I said, a universal phenomenon. You find it in different non-living and living materials, and we're just going to keep that in mind because I'm going to address now. Before I move on, I just wanted to say that you can capture this phenomenon with the same theory that I mentioned previously. So by accounting for the different macroscopic elastic properties, we can actually find solutions that explain why these cylinders purl, and we can extract information about the length of these pearls or the minimal radius, etc. So now I'm going to look at a different and seemingly unrelated function in biology, which is that of action potentials. Action potentials are these electrical signals that propagate along the membrane in our brain cells. This is actually, again, a rather universal phenomenon. You can find it in many other types of cells, not only your brain cells. You have action potentials in cells in your pancreas, in your gut, in certain bone cells, cells of the immune system. We can find action potentials in all of the plants around us, etc. And this electrical signal is classically explained by just an electrochemical theory. So basically just ions that cross the membrane, usually sodium and potassium in the classical explanation, result in transient changes in the electric potential difference, which is measured by electrode, and this signal propagates along the membrane of the cell. There's no prediction or any connection to any other type of changes, including movements or mechanical deformations of the cell surface. And nevertheless, mechanical deformations that co-propagate action potentials were identified already 40 years ago. Here you can see one of the first measurements. There's a small movement of the cell membrane, just a few nanometers, that co-propagate with the electrical signal. And such measurement was repeated by many different experimental groups and different experimental techniques. Here is another example using atomic force microscopy, and another one with a certain optical technique of coherent tomography. Now, these changes are really tiny, just a few nanometers, and very rapid, at least in neurons, just a few milliseconds. So it's very challenging to come up with a a satisfactory explanation what is actually happening here that drives the mechanical deformation. And for me, my interest is the question, are excitable cells only sensitive to electricity, or what is the role of these non-electrical changes? Are they important computationally, for example? And because these changes are so tiny, there has been several different conjectures discussing what drives these non-electrical changes. So I'll just briefly mention this. One idea was that just during the electrical signal, there are ions and water that cross the membrane and result in some transient change in the volume of the cell. Another idea was that there are different charged proteins that are located adjacent to the membrane in the cell cortex, and they respond to the change in local concentration, thus resulting in some kind of reversible conformation, maybe even a volume transition. There's a third idea that the lipid membrane itself undergoes some kind of a reversible phase transition, which results in a slight change in the width of the membrane, just a few nanometers. Or a fourth conjecture that there's electromechanical coupling at the membrane itself, which acts like some kind of capacitor. So changes in electric potential difference result in some kind of changes in the conformation of the lipid membrane itself, irrespective of any phase transition. And now I would like to take you to another example of a membrane deformation, which none of these four conjectures can actually explain, because all of these ideas formulated in different mathematical ways discuss about changes of just a few nanometers. And if we examine the mechanical deformation that co-propagate in electrical signals in a certain plant cell, as I mentioned, plants also produce similar action potentials along their membrane. The size of the deformation is larger than a few nanometers. So this is one example of a plant cell that can produce action potentials. It was highly used in the last 100 years as a model for studying the electrical activity of the cell. So you can measure action potentials propagating along the membrane of the cell. And indeed, it was demonstrated that in parallel to the electrical signal, there's a deformation of the cell membrane. So what you see here, this is the resting state of the membrane. And as action potential is induced, there was a slight movement outward with a few nanometers, and then a movement inward of the whole membrane with an amplitude of almost 100 nanometers and subsequently relaxation. Interestingly, in this example, in these cells, the time duration of the deformation exceeds the time duration of the electrical signal, even though they are completely coupled. So it was not possible to measure one without the other. So 100 nanometer, it's a bit puzzling. Where does that come from? But what we have is more of the same. It's not really clear what can we say about these movements. And so now I can take you to the work that I participated in. I collaborated with a good friend and colleague, Christian Philafer. We were postdocs together in Germany. And in plant cells, the cells are usually enveloped by a relatively rigid casing, which is called the cell wall. It is made of different polysaccharides. And Christian asked the question which led us to this work. Well, what if the membrane is really tightly pressed against the cell wall and the cell wall hinders an even larger movement? Well, maybe we could observe even something better than what was previously observed. So what we did was to plasmolyze the cell. We basically just changed the osmotic force in the extracellular solution. So water diffused out of the cell to balance the osmotic force. The volume decreased a bit and the membrane was peeled off from the cell wall. And indeed, in this case, when we excited electrical signals, we observed an even larger movements. So I'll just take you to this movie. So what we see here with the standard light microscope, the white stripes, this is the cell wall of the cell, and the black region, which is actually green in reality, this is the cell body. In the native state, the cell is fully highly pressurized and the membrane is tightly pressed against the cell wall, but because of the plasmolysis, we actually deflated it a bit. So now we can focus on a small region of the cell membrane. And yeah, and now when exciting an action potential, you will see this high movement, as you can see here, and then subsequently a relaxation. So we're talking here about few micrometers, far larger than one can expect from a few ions and water molecules diffusing during an action potential, or due to, I don't know, phase transition in the lipid membrane or something like that. So we can now choose a point and plot how this point changes as a function of time, and this is how it looks. This is called the chymograph. So the upper trace, this is the electrical signal that we measured for this cell, and this is the mechanical movement, as I said, a few micrometers, something you can easily watch with this standard microscope. And we can do even better. We can say even something else and connect that to a model. So if when we plasmolize the cells, we actually change the balance of forces that are acting on the cell surface. And if we wait enough time until the cell reaches some kind of new equilibrium, this new equilibrium is the same Perling state that I mentioned a few slides ago. And it is interesting now to observe what is happening to the cell membrane movement during an action potential as cells are kind of pearl. So I just want to show you these two quick movies. Right. So I'll just stop it here and I'll explain. What we see here is after waiting a few hours and the cell kind of reorganizes itself, so there's a dynamic movement, and we're focusing here on the region of the tether during the formation. So when I will start the movie, you will see kind of this movement as the membrane acts this surface tension forces to form purling. And when we excite an action potential, what we see is something which looks almost as a reversibility of the purling formation. And as action potential kind of moves away, we go back to kind of the purling formation. So for us, it looked as if there is some kind of reversibility of the purling process. And we can see something similar if we wait for the steady state. So this is again a type of a cell. So this is one part of the cell body. This is another one. And there's a very thin membranal tube in the middle with a diameter of just 1 or 2 micrometers, so you can hardly see that. And when we now excite an action potential, you will see an outward movement in this region and an inward movement in this region. I hope you could see it via Zoom. So just place my cursor here, so maybe you could see that. Yeah. So now we're talking about amplitudes of 30 micrometers. But when looking at it like this, we really have this idea that maybe the electrical signal drives some kind of reversibility of the purling process. And since we discussed that the purling process can be explained by this relatively simple elastic model, which is based on continuum principles without looking at the molecular details, maybe we can actually extract from these movies some information about the elastic changes. So we did that. So from the movies, we extracted traces of the cell. So the red curve, this is a trace of the cell at the resting state, and the black curve, this is a trace of the same cell at the peak of the action potential. And we can use the elastic model to find curves that match almost indistinguishably the experimental findings. So we can actually connect values of the surface tension and bending rigidity to these curves. And this is how it looks when you make the analysis. So for different values of the bending modulus, surface tension, and pressure difference, we can find regions that are corresponding to certain shapes, and we can explain the shape transformation by suggesting that during an action potential, there are changes in one or all of these elastic parameters, either surface tension increases or bending rigidity decreases or something similar. Now, we cannot resolve exactly the trajectory in phase space. We are limited in our resolution, but we can say something about the small region of parameters that these shapes are confined to. And we can extract some orders of magnitudes of these values, and there are independent ways to measure them and see that these values are indeed kind of matching. And this is where we are now. So I'll just kind of wrap up this first story. I started with a phenomenon of action potential, an electrical signal that propagates along the membrane and has nothing to do with mechanical changes. And nevertheless, one can suggest that during an action potential, there are some changes in elastic properties of the membrane, which can explain these large mechanical deformations of the cell surface with a relatively simple mathematical model. And then comes the question, well, but how can we explain mechanical deformations? Where do they come from? And this brings me to my second part of the story. So if you have any questions, please stop me. And if not, I will continue to the second part. Okay, so in the second part, I'm going to talk about sound waves that propagate within a material. We know that sound waves exist in any material. We have our best intuition about sound waves in the air, and indeed sound waves were measured within lipid membranes. So I'm going to show you some results that were taken by Matthias Schneider from Dortmund. I did my postdoc with him in Germany, and his former PhD student Shami Shrivastava. And they used a Langmuir jet trough to make measurements of changes in density and pressure within a single layer of lipids, which one can construct separating water and air. This is some kind of a cartoon of how we imagine that these things happen within this flat layer. And the question will be, well, what kind of properties do these sound waves have, particularly that lipid membranes in physiological conditions exist close to a certain type of phase transition where the kind of the tails of the molecule organize themselves from a less ordered to more ordered phase. So there is some kind of a steep transition, steep change in density as pressure increases. And the question is, well, maybe again, nature found some use for it. Maybe you have some embedded proteins that undergo some conformational change, thus exciting sound waves to propagate within the material and maybe communicate with another embedded protein or somehow also with the intercellular environment. So I'm going to show you some results about what properties these sound waves have. So they did experiments, and I coupled the experiments with a theoretical model, which is based on, again, on some hydrodynamic macroscopic principles. So just to briefly say something about it, we assume that without looking into the molecular details of the membrane, we assume that there are certain conservation laws that we cannot ignore them, like the conservation of mass of the lipid membrane, conservation of momentum, conservation of mass, momentum, and energy. And these are the mathematical equations that we used, which are formulated in the Lagrangian frame of reference. So we describe the membrane by certain macroscopic fields, density, pressure, temperature, all of them within this kind of a thin membranous layer. And the main question, which kind of drives the difference between standard sound waves that we have intuition from what we experience in the air, is the question, well, what happens if the lipid membrane is located close to a phase transition? So we are somewhere here, and then a slight compression, a local compression of the membrane drives it through the phase transition to a different type of phase. And indeed, in lipid membranes, you can find measurements that are similar to these kind of isotherms or different types of kind of pressure, density, temperature relations. And a van der Waals model is the first order approximation that can capture this kind of behavior. So we are kind of coupling these dynamic conservation laws to certain constitutive relations of the lipid material. All right, so we just solve these equations numerically and analyze their properties. And I would like to show you just briefly a few results of how do sound waves look like and compared to experiments. So these are measurements of the change in density within a lipid membrane as a function of time. So Shrivastava and Schneider, this is their work, not mine. They locally excited a sound wave, so they pushed the membrane a bit and they measured the change in density at a certain distance. When the membrane was far from a phase transition, there was a sound wave that was propagating with a small amplitude dispersing, and by the time it reached the sensor, they could hardly detect any signal. However, if they started the system very close to the order-disorder phase transition, using exactly the same excitation, they actually detected this significant increase in density followed by a rarefaction phase, a decrease in density below the resting density. And when I use the theory that I showed you, it's a rather general hydrodynamic theory, you can find the same behavior. So far from the phase transition, the density as a function of time has a certain behavior with rather low amplitude. If the initial state is close to the phase transition with exactly the same type of excitation, we have this kind of high increase in density. And this is because at the phase transition region, the membrane is highly compressible. So it's very easy to drive it through the phase transition and reach these kind of high changes in density. And it's not just kind of this switch-like behavior, which is similar between theory and experiment, but other properties. For example, one can extract the speed of sound in lipid membranes. And these are the kind of the range of speed of sounds. They depend on different parameters. For example, the viscosity of the extracellular solution or the external bath solution. And one can extract proper scales from the hydrodynamic model, and the velocity scale is related to the critical point of the phase transition. And we can either independently measure it or look at values from the literature, and the scales kind of match one another. And now it is very tempting to ask the question, can we find any example in the biological literature of signals that have similar shapes and velocities and other properties? And interestingly, when you look at action potentials, these electrical pulses I mentioned before, they also propagate along lipid membranes. They have very similar typical shapes. They have very similar velocities when you look at that. And they share many other strikingly similar properties with sound waves. So I just will give you one example among many. We can look at the response of the amplitude of these pulses to increasing the amplitude of excitation. So this is a measurement made in a non-living lipid membrane, looking at the change of density as a function of the amplitude of excitation, and the result is some kind of sigmoidal. At very small amplitudes of excitation, noise level was high and sound waves were hardly detected. But there was a certain threshold above it, the amplitude of sound waves, these are nonlinear sound waves, and the amplitude increases nonlinearly. And for even larger amplitudes of excitation, we have saturation of the pulse amplitude. From the hydrodynamic model, which accounts for a phase transition, a very similar behavior is obtained. This is very different from our everyday intuition of small amplitude sound waves in the air. And you can explain that from the phase diagram. I'll just mention briefly that the saturation is obtained because in the ordered phase, the molecules are more organized. There is the exclusion of volume, and we can no longer press the membrane above a certain maximum density. And this is one of the most famous properties of action potentials. They have a very similar sigmoidal response to excitation. You can increase the excitation amplitude and get kind of an all or none behavior. This is how it is called in the biological nomenclature. Either you get pulses that are indistinguishable from one another, or you get something which is really tiny. There is a very significant difference between these two pulses, although both propagate along lipid membranes. These are electrical measurements, and these are mechanical or measurements that are made on the density of the lipid membrane. So I just want to express that sound waves in lipid membranes are far more complex and interesting than just mechanical changes. Because lipid membranes, in many cases, specifically in biology, are electrically charged. So there's a charge density to the lipid membrane. And when we have changes in density, which result in changes in charge density, we expect to have electric fields that penetrate the bulk and thus also modify the behavior of certain charged ions that accumulate near the membrane. So there are some measurements on that, and what I did was to expand the macroscopic model and include a coupling to electricity and interactions with different ions. So these sound waves in lipid membranes, they have multiple aspects that co-propagate. There's the density change that propagates along the membrane. I discussed about that a lot. And in parallel, there's a change in the two-dimensional pressure within the membrane, which is linearly proportional to the surface tension of the membrane. So we do expect that there are changes in elastic properties during these nonlinear sound waves. There's an adiabatic change of temperature that co-propagates. This is again rather general behavior in acoustics. You compress the medium, there's some slight increase in temperature, you expand, and there's a slight decrease. And there are other changes. For example, different ions change their concentration near the membrane and their absorption and desorption from the membrane. So for example, there are different changes in the concentration of hydrogen ions, which was also demonstrated and measured experimentally. And if you break the symmetry across the membrane, there are certain changes in electric potential difference. And it's important to stress here that this kind of theory is rather general. There was no fine-tuning involved, just hydrodynamics, electricity, and you take physical parameters from the literature about the membrane viscosity, heat capacity, thermal conductivity, etc. So the shape, again, resembles the change in density and resembles what is found for action potentials. And also the scale of voltage is rather similar. So it is very tempting to think that maybe it's a coincidence, but maybe indeed these electrical signals that we have in our brain are in part not only electrical, and maybe sound is part of the mechanism. And therefore, perhaps non-electrical changes are not less important than the electrical ones. So this is something that we further pursue. There are several predictions for that. I'm just going to mention two. One is the adiabatic change of temperature. In action potentials, temperature signal that co-propagates the electrical signal was measured already 30 or 40 years ago. And this particular paper discusses how the classical electrical model cannot really explain that. So this was something that remained a puzzle, but nobody really further pursued it since then. But with the idea of sound, this is rather natural. This is the signature that we get. The other one is this tiny deformation that I mentioned at the beginning. So for high curvatures, we expect to have these kind of elastic changes in surface tension and rigidity. But even for almost flat membranes, when you drive the membrane across a phase transition, this is well known that the molecules slightly reorganize themselves. And there's a tiny change of 1 or 2 nanometers in the width of a lipid bilayer. And therefore, again, this is something connected to a conjecture that was expressed way before my work. But this is, again, something that comes out from the theory. And there are several other predictions, but I think I will just take advantage of that and move on to the third story. I'll just briefly summarize this part. If you just think of a medium where there's a phase transition involved, sound waves are an emergent phenomenon for any material. Specifically, if there's a phase transition in the region, and you can have the same thing also for water in the gas-liquid transition, sound waves will have certain nonlinear properties. For example, the sigmoidal response to excitation amplitude. This is unavoidable. If your medium of interest is electrically charged, then you can expect that in parallel to the changes in density and pressure, there will be associated electrical and potentially also chemical changes of different kind of particles that are involved. And finally, I showed you that there are some striking similarities between such nonlinear sound waves in the lipid membrane and action potentials that are electrical signals that also propagate along lipid membranes in biological cells. So any questions so far, or I will move to the third part. So the third function property of a material I would like to mention is that of an abrupt or steep transition in the conformation of charged polymers as a result of different environmental conditions, and we can find that in many different polymeric systems. And this is one example where you take a non-living polyelectrolyte gel and change different properties. So I'm interested in the competition of mono and divalent cations, which is something natural to expect to happen in living cells. So this is an experiment where the volume of the gel was measured, normalized by the volume of the dry material as a function of changing the concentration of the divalent salt in the external bath solution while having a fixed concentration of the monovalent salt. And so in this case, these gels are highly swollen. I think we discussed that a bit with Guy before the talk started. So 99% of what you see here is water, and just less than 1% usually is just solid material of the negatively charged polymers. And as we increase the concentration of the external bath solution in the salt, and there's a gradual decrease in the gel volume, but there's a critical value of concentration where suddenly there's a steep decrease in volume as these kind of polymers collapse and phase separate from the liquid, resulting in an almost dry gel. So here in this case, 50% of the material is solid, and just 50% is water molecules that are trapped inside. So you can find this behavior in several different non-living gels, but interestingly, you can also find similar behaviors in living systems. This is one of my favorite examples. The blue rectangles that you see here are measurements made of gels that are made of DNA molecules. And the DNA molecule is significantly different than the polyacrylate that was used in the upper curve measurements, but nevertheless, the macroscopic response of the gel volume to the ionic competition is very similar, same orders of magnitudes of volume, of transition, of critical calcium concentration. And you can find the same. So we know that the gels the DNA that lives inside the nucleus, it kind of expand and compact as a result of interactions between different multivalent cations of different types. And there are several discussions in the literature that perhaps this opening and closing of DNA can be connected to these kind of fundamental principles taken from standard polymer physics. A similar behavior was found for different types of secretory products. So there are different cells in our body that secrete gels. For example, the most famous one is mucus that we have in our lungs and in our gut. And the mucus is compacted inside cells using calcium ions that are divalent. And when the vesicle spills the mucus gel into the extracellular solution, there's a high concentration of sodium. There's a competition, and the mucus gel rapidly swells. So again, rather similar physical description, even though the molecular details are of course different. And you can find, again, some similarities in the properties in pectin gels that are abundant in many land plants within the vascular tissue. So changes in the salt concentration of the fluid, which is being pumped by the roots, can modify the state of the pectin and thus controlling the rate flow of the water. There is now a new sexy field in cell biology or biophysics, which is called membranous organelles, different types of structures inside a cell body, which are presumably just different types of charged polymers that phase separate from the cytoplasm, from the cell solution. And yeah, and there is some other measurement of the electric potential, which is connected to competition of mono and divalent cations. So different types of unrelated systems, what connects all of them is the idea that you have a negatively charged polymers interacting with water and monovalent and divalent ions. So what we are hoping is that by looking at this non-living gel, we can maybe find some kind of mathematical formulation which can describe this competition and thus giving us a framework for the far more complex biological scenarios. And it is interesting because even this very simple system, it's just one very simple type of polymer, water, and just one or two types of salts, so few ions and negative ions and positive ions. This is by itself a rather complicated system. When you want to do some molecular dynamic simulations, you can hardly capture equilibrium behavior because you can only make predictions over nanoseconds or maybe microseconds if you really are willing to wait a lot. But even if you want to write down continuum mechanics, continuum dynamic simulations, the models are rather complex because you have to account for the fluid as one material and the solid cross-linked polymers as another one, and there should be another field for the different types of ions, etc. But this is what we were trying to do. These gels, even though they are non-living and synthetic, they produce some rather complex behavior. I don't want to touch that. I'll just skip into the model that we calculated, and I'll spare the equations. I'll just show you one result. So what we can do, is we can write down, again, universal equations. So conservation of mass, momentum, and some description of the interaction, the phenomenological interaction between calcium ions and the polymers versus sodium ions and polymers, and maybe also accounting for pH. And from this model, we can extract different types of behavior. For example, here I'm showing you how the volume of the gel changes transiently from a non-equilibrium state into an equilibrium state over time. And for different values of the divalent salt in the external bath solution for a fixed monovalent salt, there will be different behaviors. You can have a swollen gel or a collapsed gel, and the gel does not, according to the theory, does not collapse immediately. There's some transient time of water diffusing in, and then another transient time where calcium ions adsorb onto the polymers and resulting in this kind of phase separation. And interestingly, when you compare the model measurements to the model results to measurements in non-living polyelectrolyte gels, you can find a very similar behavior. So the scales of the volume and time and response to different calcium concentrations are rather nicely portrayed here. So we can extract from the theory some information about different time scales of water and calcium absorption, etc. So I think I will wrap up my talk here. My take-home message is that even though living cells are very complex and the molecular details are so important in each and every function, we can hope to drive towards some kind of a quantitative, mathematically formulated description if we focus on certain aspects that are more universal and are seemingly connected to the behavior of non-living materials. And then we could find our first order mathematical description, and later we could expand that to account for the important molecular details. And I showed you these three examples: the Perling instability, which is abundant in soft materials, sound waves that drive a local phase transition, and a volume transition in charged polymers driven by competition of different ions. Right, so thank you for listening, and if you have any questions, I'll be happy to take them. Thanks.
Thanks, Matan. Thanks for the interesting talk. Any questions for Matan?
I have a question. I'm in the meantime, if others are thinking, may I ask?
Yeah, go ahead.
Okay. Matan, when I'm talking about a volume transition and behavior of polymers or polyacrylamide, didn't you think that, like, if you would consider clays, low silicates, which are basically polyelectrolytes, negatively charged polyelectrolytes, and it's now that they may demonstrate a transition of flocculation behavior at certain concentrations of multivalent cations. So don't you think that might be some similarity in behavior? And if so, I mean, there are quite many theories were developed to explain flocculation of negatively charged clay particles in the presence of multivalent cations.
So I'm not. So you're talking about clay, right?
Yes.
Okay. So first, I'm not entirely familiar, but I don't see a reason why not. I mean, so I did not show you the equations I used for describing this, but again, I did not invent the models of volume transition. So the model I used is based on the most classical Flory model, so Flory-Hagens model, which was developed already 80 years ago. I did not use anything new. I used the most appropriate model one can think of. The only thing I did was to couple this Flory model, which usually focuses only on equilibrium states, is to couple it with the dynamic equations of transient changes, which is again based on classical hydrodynamic principles. So you start with conservation of mass momentum, and then you can follow the dynamics of the water diffusing in and the salts diffusing in, and accounting for how they interact with the polymers by using the phenomenological Flory parameter. Again, this was done 30 or 40 years ago. The main difference in this work that I showed you was to account for a relation between the classical Flory parameter and the interaction of ions, which this is usually, you don't find that much in the literature. And if you know about something in clay, I mean, maybe I should look about that, because I was not aware that clay are negatively charged polyelectrolytes, and certainly, I think usually probably clay is very highly cross-linked, right? Because it doesn't swell much. Or...
They do swell, they swell perfectly, depending on the cation. That's exactly the point. And there are a lot of physical modeling. How do they swell? How do they flocculate? And it was basically classically explained by electrostatic models, by considering that cations neutralize charges on alumisilicates, allowing them to one plane to each another, and then van der Waals interactions might lead to aggregation and flocculation. So there's a long story, like, you know, probably.
No, this is a wonderful comment, and thank you.
At the same time, when Flory was developing his model, and it was probably 40s or 30s, and probably the same time was a physical modeling of clay behavior.
Yeah, but I just want to add, you mentioned, Matan, cross-linking. In clays, there is no cross, which is a prerequisite for the water-absorbing polymers. This is the thing. And another thing is with clays, unlike the polyelectrolytes you work with, you can eventually end up at a situation where you get dispersion of the clay particles.
Because they are not cross-linked, so they can flow.
Yes, exactly. You can, I mean, if you apply, I mean, if they absorb enough water, eventually they disperse, which will not happen with the
so this is really interesting, because I mentioned just briefly, there's one very, I don't know, it is now highly explored in the biological or biophysical literature of membraneless organelles, where there are different types of charged proteins and RNA molecules that are not cross-linked, but are just phase separating from the liquid or disperse in it. And then when they disperse, they can really flow indefinitely within it. So that's an interesting comparison. I wrote that down. Thanks.
And I have one more question, actually, very general, because I mean, I admit that I don't really follow everything you said. Many things are far from me, but when you're talking about effect of surface deformation of membranes and their action potential, so, and you said it's reversible, right? So you're applying a mechanical model which includes all reversible elements. Okay, but may show some hysteresis when you are have plotting surface deformation against potential going up and down. So is it really reversible in the sense that you may see, if it is possible to get such curves, do they coincide or not? is there any meaning to see a surface deformation as a function of potential in two directions?
So your question is the deformation goes back to the same initial state?
Yeah, you're applying a reversible model, you said, but I'm trying just to convert with vision to something which I'm more familiar. Is it possible to have a plot of surface deformation as a function of action potential? And if you're increasing one and you have changes in surface deformation, and then you're going back, may you have which kind of the curves describing surface deformation is a function of the potential, and if they're coinciding depending on direction?
So you mean actually hysteresis, Michael?
Exactly.
Yeah. So, I mean, hysteresis was found in different examples. I'm not sure if for the case of deformation it was ever explored. I don't know how easy this can be done. But in terms of action potentials in general, or driving the membrane by increasing the external electric stimulation, you can see some hysteresis effects.
And
I never looked into that.
In that case, it becomes non-reversible or not so simply reversible.
Yeah, no, that's a very good comment, and yeah, I really don't know in that respect. I mean, when you just use the sensors that you have, you consider the membrane is at a certain height, and then during the electric signal it moves up and down and then goes back to more or less the same surface. But does it really mean that it's reversible, or were there some internal changes that we are not aware of? I don't know. It's a good point. Yeah.
Okay. Thank you, Matan.
All right. Thank you.
Okay, so if no more questions, we are already at noon. So I would like to thank you again, Matan.
Thanks for the invitation.
Very much.
All right.
And see you next
Matan.
week.
Thank you very much.
Matan, if you want to further discuss polyelectrolyte shells and water absorbing, then I'll be more than happy.
I think one day when we are after the wave, I will come visit Udi and I will come visit you too. Thank you. I'll do that.
Great. Looking forward to that.
All right.